So let us discuss with the second law of Mendel, which is the law of segregation. The law of segregation. Segregation means separation. So what does this law state? The two alleles of a gene. The two alleles of a gene. We know that a gene exists in two alternative forms which are called the alleles. So these two alleles of a gene separate during the gamete formations. They will separate during the gamete formation so that each of the gamete will have only a single allele in it. So let us understand this through a monohybrid cross. So a monohybrid cross is a cross between parents which will differ with respect to one character. So here, okay, I have made a cross, okay, between two parents which differ in their height, which is the character of difference. So we have taken a tall plant, okay, as a male plant and a dwarf plant as a female plant, and when a cross is made, what is happening if you see, okay, so this is the male gametes, these are the female gametes on the top, what I have written. Okay, the male gametes and female gametes, what I have written on the top, they represent actually haploid status. That is, see, when the gene is there, when both the alleles are like this in the parent, during the formation of the gamete, they get separated from each other. So we have two gametes, okay, which have one allele each, capital T and capital T, from the male parent, and we have two alleles, okay, small t and small t, coming from the female parent. So during gamete formation, okay, we know that gametes are usually haploids. They are haploids only. So from a diploid cell, we get haploid gametes. So you know humans have 46 chromosomes. This is the diploid cell. So the gametes, whether they are the ova or the sperm, they will have only 23 chromosomes, which is the haploid number. So this is called the diploid number. So the same principle, okay, applies here also. During gamete formation, these are the gametes, okay. What is happening? The alleles will separate from each other so that each of the gamete will get only a single allele. Okay, so this type of a board when you draw, this is called as a checker board, or it is called the Punnett square. Okay, it is called the Punnett square. Okay, Punnett square it is called. Okay, or it is called the checker board. So in this, what you will represent? The male side. Okay, this side, what are the male gametes you are representing? So in this box, you will write the male and female section there. The male gametes you will represent this side, the female gametes that side. And when you have to fill in the checkerboard, okay, you have to just put the two alleles together, capital T and small t, capital T and small t, capital T, small t, capital T and small t. So like this, what we are getting out of this is the F1 generation, the first filial generation. So as we already know, after a cross, immediately the first generation of individuals, whomsoever you obtain, those are called the first filial generation individuals, the F1 individuals. So these are all the F1 individuals. The status of all the F1 individuals in this cross is phenotypically all of them are just tall. All are tall. All are tall. Then usually in a cross, okay, to make a better study, what they will do is they will also generate the F2. Okay, they also generate the F2. So how the F2 offspring are generated? By selfing F1, we will get F2. What do you mean by selfing of F1? See, F1 individuals all have the same genotype, that is capital T and small t. Selfing means both the parents, okay, when you take them with the same genotype, it is called selfing. So here I have shown selfing of F1, that is capital T small t, capital T small t. Either of the parents are having the same genotype. So upon selfing, okay, the F1, we get the F2 individuals. So again, you can develop the checkerboard here, wherein, okay, I have written the male gametes this side, the female gametes on the opposite side. So here the male and female gametes, if you see capital T small t from the male parent, female parent also capital T and small t we will get. So you have to just okay, add on these to put them in one box, wherein you can fill all the boxes like that. So these are called the F2 individuals. These are called the, okay, after selfing the F1, you are getting the F2 individuals. So this is the F2 individual section what we got. So out of all these F2 individuals, okay, if you see the phenotype, how they will appear, okay, if you see capital T and capital T will be tall only. We know that capital T, whenever it is present, it is a dominant allele and it will reflect itself. So this is tall. When capital T and small t are present, then also it will be a tall plant, because small t is a recessive allele which cannot express itself. According to law of dominance, when two contrasting alleles are present, the dominant one will be expressing itself. So here again the small t and capital T, okay, when you write them together here like this, so it is also a tall plant, and the small t and small t together when you write, it becomes a dwarf plant. So after you write the checkerboard, we write what is called the phenotypic ratio and the genotypic ratio. So what is this phenotypic ratio? So out of the plants, okay, that have come here, how many of them are of a particular phenotype? So how many of them are tall? How many of them are dwarf? We are writing here. So phenotypic ratio is 3:1. That is 3 tall plants is to 1 dwarf plant, you write. And then the genotypic ratio. What is this genotypic ratio? How many types of genotypes do we observe here? Capital T is one genotype, capital T small t is another genotype. So these two are of the same genotype. We have one capital T, two capital T small t, and one will be small t. So our genotypic ratio is 1:2:1. One is capital T, one is small t, two is capital T and small t. So like this, you will be writing the phenotypic and genotypic ratio for a monohybrid cross, and whenever in the exam they ask you to explain, okay, this law of segregation, you have to explain it through a monohybrid cross like this. So okay, on the whole, what this law is stating is the alleles which are present in the gene pair will separate during the gamete formation. That is, let us suppose if two of your friends are sitting together for an English class, okay, you are assembling in A section, let us suppose, okay, so though you belong to different sections, one girl from A section, one girl from B section comes together and they are attending an English class, let us suppose. So let us compare that, okay, these two as alleles, that is A section girl and the B section girl are the alleles sitting together. So after the bell is rung, okay, which can be compared with the gamete formation, both these alleles will separate. That is, both these friends who were sitting together, they go to their respective sections as a single person. A goes to A section, B goes to B section. So this is what is happening during the law of segregation. Okay, so hope you understood through this particular example. Okay, it is quite easy. Any of the doubts, whatever you have, you can post in the comments. I will surely help you out, whatever it is. Okay, so hope you followed. Thank you.